All right, so in the interest of time for everyone, we'll get started. So my name is Amy Wong, and I will be providing this overview of the RFP for accelerating the development of point-of-care molecular diagnostic platforms. We will be saving the Q&A for the end, but you can, throughout the presentation, see in the Zoom toolbar, there's actually a Q&A button, and please submit your questions there and not to the chat, but to the Q&A. And we have moderators right now who are will collect the questions for the very end. All right, so let's just dive right in. So just quickly, the summary, overall summary of it is that FIND is aiming to accelerate the development, manufacture, and launch of a point-of-care, multi-pathogen molecular diagnostic platform for use in LMICs. And for everyone's, to get on everyone's radar is that this RFP is funded through Access to COVID-19 Tools Accelerator, also known as the ACT-A, in their diagnostics pillar. And a total budget envelope of 25 million US dollars is available to selected manufacturers, specifically to enable development of a platform that meets a set of predefined key product requirements. And I'll be going over a little bit of that coming up. And part of this RFP is that the suppliers will have to commit to supplying an affordable product. And again, I'll review that in the later slide, as well as submit a dossier for the WHO's pre-qualification program by the end of 2022. So the product that we specifically are some of the key characteristics of the product that this RFP is aimed for is this product is meant to be used as a differential diagnosis of respiratory pathogens, including COVID-19. Other respiratory pathogens that are of interest to us are flu A, flu B, RSV, strep pneumoniae, and mycoplasma pneumoniae, and tuberculosis. This product also is intended to be suitable for use in decentralized healthcare settings. And again, that's more specified in the product requirements. The product is meant to be a sample to answer, meaning that it requires minimal user training and minimal sample preparation. And again, it is meant to be affordable by low to middle income countries. And again, I'll review that and what that means in another slide. So the types of activities that are supported by this RFP are primarily pre-launch activities. So for example, and this list is not extensive, but are key examples: assay development, verification of performance or validation of a multi-pathogen respiratory panel. Or if, for example, your system currently does not meet the requirements, modification of that assay or instrument to meet those requirements is covered by this RFP. Some clinical validation, pilot studies, usability studies. Also optimizing your manufacturing capacity or increasing or providing investments into your manufacturing capacity so that you are able to serve the volumes required in LMICs. Also some financial or logistical operational support for in-country registration, as well as trials that will meet the regulations of WHO pre-qualification program. And again, this is targeted so that, again, this product is marketed in lower middle income countries. Also, some of the preparation for launch, for example, market planning launch strategies, commercialization strategies, forecasting, for some of the pre-launch activities that will ensure that the product is marketed in these countries will also be supported. So the timeline is very condensed. We understand that we are asking for clinical validation in 2022, followed by submission of a dossier to the WHO pre-qualification program by the end of 2022. So with that, as you know, the RFP is closing September 3rd, which is just under a month away, upon which we will undergo review as well as potential interviews and presentations with shortlisted candidates, with the hopes that selection of finalists will happen somewhere towards the end of 2021, with ultimately contract negotiation to begin and awards to be decided in early 2022. So ideally, the suppliers will have January through September of 2022 to undertake the R&D activities described in your proposal. And again, the end goal is for regulatory submission to the WHO pre-qualification program by the end of 2022. So the suppliers or the applicants will be judged based on some criteria that are listed in a document called the assessment matrix, which is available for download on the website. And within the assessment matrix, there is actually 3 sheets. So the assessment matrix is an Excel spreadsheet. So there are 3 sheets. The first one is the technical assessment. The second one is the business assessment. And the third one is the alignment criteria. And again, I'll go over a little more specifically what these are. But the technical assessment is where the product requirements that we are aiming this product to meet are listed. So additionally, there will be an applicant presentation. And that will also, we ask that all applicants use the template that's also downloadable off the website. And then shortlisted candidates will be requested to make a follow-up presentation where they will answer a set of questions that will be sent to them. And then in parallel to most of these selection process will be some due diligence, which will be contracted to an external organization to provide some background information on the shortlisted candidates. So the selection process will take place over mostly three stages. So the first stage is that all applicants will be screened against some eligibility criteria, which are things like, bankruptcy or criminal investigations. Some of this is detailed in the RFP and one of the appendices. Also, applicants that are considered out of scope will also be eliminated from the pool. And out of scope applications are ones, for example, that might be serology-based assays, rapid tests, or, for example, just reagent manufacturers without an instrument. And again, more of this is detailed in the RFP text, which you can download. So after this first screening, there will be a long list of candidates. And then that long list of candidates will be scored based on their performance in the technical assessment, where each of the product requirements will be scored. And again, I'll go over that. As well as a score in the business assessment, which evaluates some of the organizational strengths. And again, I'll go over that, as well as the information that is provided in the applicant presentation. So everyone should make sure that they read the text of the RFP to see what information we would like to have within the applicant presentation. So an internal review panel with members of FIND will score each of the candidates in those 3 areas, and then the scores of the reviewers will be averaged. And then we will down select to approximately 15 shortlisted candidates for stage 2. And stage 2, the main difference between stage 2 and stage 1 is that stage 2 will have an external review board, which will be made up of a wide range of stakeholders that range from those who are experienced in technical R&D to all the way down to implementation and delivery of a product. Again, the external review panel will look at the overall fit. So not just, they will have the scores from the technical assessment from stage 1 that will be provided, as well as the business assessment. They will also have the applicant presentation. And then shortlisted candidates, again, will be asked to answer some follow-up questions in a live presentation. So the external review panel will review the scores from stage 1, as well as the follow-up presentation, to again score the candidate, and the scores will be averaged across the review panel. So I wanted to talk about just the layout when you open up the Excel spreadsheet and click on the technical assessment sheet so that people can understand how to read it. So for those of you who are familiar with more of a detailed product requirements document, the goal was to mirror that to help you understand the requirements that we were asking for. So you'll see that across the top will be the category of the design requirement, the market need, and then detailing what the optimal specification and the acceptable specification is. So in this example, where the design requirement is asking for storage conditions for the assay kit, there's an optimal design specification, and then there's an acceptable design specification. And we are asking you as the applicant to fill out the column titled existing specification, as well as evidence or data supporting that claim. So we would like you to tell us about your system as it exists today. So for example, if you don't meet the optimal design specification, but you meet some parts of the acceptable design specification, please write down what storage testing you have done and what you think your storage shelf life is. And then what's critical here is that in your presentation, in your applicant presentation, we would like you to explain to us for those specifications which you do not meet at least the acceptable design specification, include that in your R&D plan of how you will modify them. And it doesn't have to be on all of the specifications, but primarily the ones that are noted with a 3 under the weight column. So here, storage conditions is weighted as a 2, meaning that it is important, but it's not the utmost critical ones. The critical ones will be denoted with 3. And for your information, you'll see that there is a score column, there's a weighted score column, and there are some scoring guidelines. These columns are for the reviewers to be using. And so the weighted score is basically the weight multiplied by the score, and the score usually can range from 0 to 3. And so you will see that some specifications have a heavier weight than others. So again, in your applicant presentation, as well as your R&D plan, you should describe how you plan to modify your existing specification to meet either the acceptable or the optimal or something in between for those specifications denoted with a 3 in the weight column. And then there's one particular design requirement that I wanted to provide a little bit more context on because I understand it could be a bit confusing. So this RFP is specifically looking for a multi-pathogen multiplex platform. However, we understand that multiplexing might be part of someone's R&D plan if they're able to singleplex already. So for example, they might have a standalone flu assay or a standalone COVID assay. So we wanted to provide points in scoring for those who either have an extensive test menu so they can singleplex many of the key pathogens, as well as we wanted to give points to those who also were able, who have demonstrated the capacity to multiplex. So for example, they had a COVID flu RSV panel assay. And so you can see in the first row here, multiplex capacity, the optimal design specification is that it can detect 4 or more pathogens simultaneously from a single sample. And of course, the acceptable design specification is that you can only detect 1. But you can see in the scoring guidelines that points are given for how many pathogens you can detect in that single assay. And you can also see in the weight column that multiplex capacity is denoted with a 3, meaning that is a higher priority for us and should be addressed specifically in your applicant presentation and R&D plan. Whereas in the second row describes systems that might have, again, an extensive test menu, but not the capacity to multiplex currently. But we still wanted to provide the opportunity for those applicants to score as well points. And again, if you can only singleplex currently, please describe how you will multiplex some of the key pathogens in your applicant presentation. And we have received a question by a few applicants already about whether or not the supplier has to already have a COVID-19 assay. And although COVID-19 is obviously a large focus of this, it is not a prerequisite. So if you have not developed a COVID-19 assay, but you have perhaps other respiratory pathogens, that will also be scored and provided points for. So I also wanted to go over what would be the second sheet in the assessment matrix. And this second sheet is called the business assessment. And so here there are some broad categories or criteria in which we will be evaluating your organizational strength. So again, you're asked to fill out the column titled Evidence and Supporting Information so that we are able to evaluate your organization. And again, we will be having due diligence run in parallel with an external organization to verify some of the claims. One of the criteria that I really would like to provide some clarity on are the COGS. So again, a large focus of this RFP is to have affordable molecular multi-pathogen platforms available for LMICs. And with that, we would like to again ask applicants, and there's a specific place on the applicant presentation template for you to provide this information, is for you to describe your production COGS forecast over 5 years, where year 1 is 1 million tests with 200 instruments, growing to 10 million tests and 1,000 instruments at year 5. So our goal, our target COGS is $5 if you can only detect 1 pathogen, or $10 for a multi-pathogen assay. at 10 million tests per year. And the target COGS for the instrument is $10,000 at 1,000 instruments per year. We've had some questions about whether or not this is a prerequisite or will be. What if the candidate cannot meet that strict requirement right now? And again, what we are asking for is clarity on what you think your projected COGS will be. And then in the applicant presentation, please describe how this R&D funding may potentially drive those COGS down. For example, we could invest in a manufacturing capacity, invest in some materials validation or testing of the manufacturing line. The goal is that the funding from this RFP can help drive the COGS down. So again, if you do not feel that you can meet the target COGS at $5, please provide a reasonable plan for how you can drive it down to $5 or $10, depending on whether you will do a single plex or a multiplex assay. So lastly, there is a third sheet, and it's called the alignment criteria. It's really provided for your reference. This is going to be completed by both the internal as well as the external review panels in 4 main areas. So the value proposition and impact, the value for money, technical merit, and organizational strength. And so some of the scoring considerations, you'll see the questions listed, are the types of considerations. It's not an exhaustive list of how reviewers will judge your application. And the scores, again, across the reviewers will be averaged across the reviewers. So the applicant presentation, this is, again, there is a template that is to be downloaded along with the assessment matrix, and please provide, please use the provided PowerPoint template to provide additional context and data that's requested. It should be no more, ideally, no more than 25 slides. And please, when you upload your documents, please make sure you upload the presentation as well as the assessment matrix. So the key information, and it again detailed in the full text of the RFP, some of the key information that I wanted to highlight is, first, we would like to know is your current system's alignment with the product specification, product requirements. So for those design requirements that are denoted again by the 3 in the weight column, please provide evidence of your current system's performance. And if it does not meet the acceptable or optimal specification, a reasonable plan of how you will achieve that. And as part of that, please provide a roadmap, including the proposed activities for your system to meet the design requirements. And then a timeline of those activities so that we are able to ascertain whether you can meet the goal of clinical validation in 2022 and submit and dossier to the WHO pre-qualification program by the end of 2022. And again, as I mentioned before, there's a specific area in the RFP where we ask you to describe your proposed pricing model. We would like applicants to consider a COGS-based pricing model, and where you demonstrate what you think your production COGS will be, again, over the forecasted time and volumes that I mentioned in the previous slide. And your expected business model, for example, are you going to be a capital purchase or reagent rental? And again, the slide also details some other areas around transparency of the price that we hope to work with suppliers on. And some of the transparency includes expected distributor markups, as well as internal markups to cover your expenses. And again, it's detailed a bit more in the RFP text, as well as the slide PowerPoint presentation template. So again, as many of you have already, please submit questions to the email address listed here. It's also in the text of the RFP. Questions will be accepted until August 26th. And all the Q&A that we've received, we will post within 24 hours on the website. So in case you do have questions, please refer to that first before to see if we've already answered your question. And again, when you submit your application, please upload your assessment matrix, which you have filled out in the columns that we've discussed here today, your completed applicant presentation, as well as some supporting documents to this email address. And the types of supporting documents that we will accept, we will not accept all and any, but we will accept things like package inserts of relevant assays or various regulatory certificates. And again, this is detailed in the RFP text. And so applications without the completed assessment matrix or the applicant presentation will not be accepted. All right, so with that, we will now go to the Q&A section. So let me just take a look here. We have a question: multiplex panels with enough sample input to get LOD is quite difficult with the computer primer probe interactions. So yes, we understand multiplexing is a difficult challenge, and we recognize that it would be very difficult to get a significant number of pathogens, upwards more than 3. And so we would like for you to describe how you will do it in your R&D plan and whether or not you have any data that's existing. There's another question: would it be acceptable to use commonly available standard PCR instruments, or is it required to create a bespoke instrument? So the goal again for this RFP is to have a primarily sample to answer instrument. We recognize that some of them may rely on upstream extraction processes followed by amplification. And again, it's not that it is ineligible. However, we would like to know from your R&D plan again how you plan to make this suitable for use in decentralized settings where minimal user training is a key requirement, as well as minimal sample prep is a key requirement. So if your existing platform may consist of an upfront sample extraction followed by amplification following, for example, we would like to see how you plan to either integrate the 2 instruments or provide some way that sample processing is no longer onerous on the user. Another question is, will procurement activities be conducted by this RFP? So this RFP does not cover procurement. So we are actively discussing with other ACTA partners who are involved directly with procurement and delivery of product to ensure that there is a smooth transition between where this RFP ends and where the rest of the active partners need to pick up. Another question, can we get a list of LMICs? Yes, in the RFP text, there is a reference to the World Bank list. And so you can take a look at that reference. Another question is, what is the deadline for submission? September 3rd, which is a Friday, I believe. Another question, can we get respiratory pathogen wish lists? Yes, and that is in the RFP, and they include COVID-19, flu A, flu B, RSV, a couple of bacterial pneumonia, and tuberculosis, TB. Another question, will the budget be split among the 2 to 3 eventually chosen projects? So the exact award amount and how many applicants we end up awarding will vary. So certainly can be at least 1, and most likely somewhere between 1 and 3. It will not be split equally. We split based on the your request and funded activities that you're requesting for. How much of detailed information will be disclosed to external reviewers? So all the external reviewers, and if the concern is about confidentiality, we will have CDAs in place with all of them. And all of the information that you provide, the assessment matrix, as well as your presentations and due diligence, will be provided to the external reviewers. So another question, so can you provide some guidance on the methods and controls to be used for reporting LOD performance? The FDA comparative testing demonstrates a large variability between claims, LOD, and FU and objective comparative testing. So I think we will be following, so the FDA's claim for, or what I've seen in terms of how to do an LOD study, I think we'll be following that for analytical sensitivity, but we understand that there's going to be a clinical sensitivity. And so please describe how, so when you state your LOD, for example, in the assessment matrix, please describe in some brief detail about how you were able to achieve that LOD, whether that was with comparative samples or spiked samples. Another question is multiplex single tube reaction for all these pathogens are expected? No. So again, we recognize that having 5 pathogens in one tube in a sample to answer point of care platform is a tall challenge. And this is why the scoring enables you to demonstrate either somewhere between 1 and 5. Ideally, again, the acceptable specification is 1, with the optimal and ideal one being 5. But again, if you note in the scoring in the assessment matrix, we do have a preference or a higher weighting for those who can multiplex. Is there a maximum budget that can be requested? Probably not more than the actual full envelope. We will unlikely fund just one with the full envelope. But the ask has to be reasonable. So if you notice in the business assessment, or not the business, the alignment criteria, one of the questions is whether it's value for money. So as long as, please just justify your ask. How is the confidentiality being assured? So we will have CDAs in place. All the materials will be held internally and will not be open to the public. So we can provide copies of the CDA if you would like to review it, if you the ones that we do provide reviewers. Just send us an email if that's something you would like to review. Another question is one single multiplex panel requested that includes, okay, so I guess. So again, optimal ideal would be, of course, let's detect all the pathogens the key pathogens we've discussed in one tube. We recognize that is a very difficult challenge, very difficult technical challenge. And so we will give points based on how many you can multiplex together. And you'll see that in the, again, in the technical assessment. Any other questions? And again, this webinar is recorded, so you can refer back to it. You can also always send email questions back to us, and we will try to respond to you within 24 hours and post also the question and the answer on the website. All right, so it looks like we do not have any more questions. Thank you very much, everyone, for either early morning, late evening, wherever you are in the world. And we look forward to receiving your applications. All right, bye.